Concentration gradient. Okay. Concentration gradient. It's all off the cuff. I'm freestyling. All right, so these blue dots are water. That's a big water molecule. All right, and red is going to be sugar. Okay. So we have 3 different regions. We have a high concentration of sugar here, a medium concentration of sugar here, and a low concentration of sugar here, and no sugar here. So sugar has the natural tendency to diffuse, which means that it wants to move with its concentration gradient. So its tendency to move, let's say it had 4 options. So its option to move here from here to here is going to be, the concentration gradient is not very steep because you have, a very high concentration here and you have a somewhat high concentration here. Now the concentration gradient between here and here is a very steep concentration gradient because the difference between this high density of sugar here and here is huge. So this is going to be high concentration gradient. Oh, I'm all caps, huh? And then you could say, relatively low concentration gradient. Why am I using, I'll just spell out concentration gradient. Okay, does that make better sense? Usually when things are moving against the concentration gradient, so let's use that as an example. So let's say we'll scratch this. We'll create new. Let's use a semi-permeable membrane as an example. So here's a semi-permeable membrane. We're just going to use, we'll say this is a cell, and water can move through freely. So water can move, through this semi-permeable membrane. We could say this is we'll say that's the lipid membrane, the lipid bilayer, or the cell membrane. Now, if you had, let's just say this is sugar inside of your cell. Okay, so you got lots of sugar inside of your cell because you just ate a huge meal and you released insulin, and that caused you to uptake all the sugar into your cell. Now, normally this sugar can't diffuse, so let's make them bigger. Let's make these, okay, that's not going to work. So these sugars, can't just diffuse through, they have to be transported in through a protein. And this is a real life example too. Or a real life example. This is something that's going to happen in your body too. So you got sugar inside of the cell and sugar outside of the cell. Now the gradient, and we'll draw a little protein too. Here's a transport protein. So the gradient, meaning the direction that sugar wants to move, just using the basic principles of diffusion, is this sugar really wants to move outside of the cell because there's less of it outside of the cell. However, we would probably prefer more sugar in the cell than outside of the cell, right? And so what we do is we spend ATP to move sugar into the cell against its concentration gradient. You can think of concentration gradient kind of like what would nature allow to happen? That's its gradient. So the gradient for sugar would be to move outside of the cell, but in this case, you're moving against what sugar wants to do, and you're going to move it into the cell. And so that's going to require ATP, and that's going to make it active transport, moving against its gradient. And the same thing would go, I mean, you could view this differently. You could view this as the proteins in the electron transport chain move the protons into that intermembrane space against their concentration gradient. That intermembrane space has a very high concentration of protons, and what you're doing is you're using that potential energy of that movement of an electron from protein to protein, or from electron acceptor to electron acceptor, to shunt those hydrogens into that intermembrane space against their concentration gradient. Okay, so we have a question. If there were a high concentration gradient, it would move more quickly. Yes. So if you had a high concentration gradient, oh, I wish I could go back. I didn't save it. But if you were to go from a high concentration of sugar to a complete absence of sugar, it will move very fast. But if, for example, let's say there was no membrane here, if you were to move from this region of high concentration of sugar to like this, moderate concentration, it would move slowly. It would move slower than it would if there were no sugar there. Okay, so Judah asks, does diffusion happen because of a protein or an enzyme? So diffusion itself will actually never happen due to a protein or an enzyme. So in this case of moving this sugar through here, this is going to be transport. So anytime you see diffusion, that's just going to be passive. That's just going to be the things naturally moving from one place to another. When we say transport, that means we're using some kind of channel or some kind of active process to get something somewhere. So like this sugar wants to diffuse through, but it can't because you got this semi-permeable membrane. And so we have these channels that transport the sugar through. Okay. Good questions. Those are really good questions. So I'm just going to throw out some hints for you guys. Know how the inhibition of an enzyme catalyzes a reaction between a competitive inhibitor is going to be different from a non-competitive inhibitor. So the answer to that, can I draw this? Yeah, I can draw this. I don't know if it's a waste of time to draw though, because it's, okay, so here's an enzyme, and actually, how do I make the erasee bigger? Okay, I'll just do some honest work here and no shortcuts. Okay, so let's just say that you got a triangle shape or something. And then your substrate, we'll make the substrate a different color. Make the substrate orange. So this is the substrate that wants to move in. And, afterwards, your product is, we could just say like two smaller triangles, I don't know. So that's your end result. So here's your enzyme, and we can write enzyme. And then here's your substrate. You can use that synonymously with reactant. And then here's your product. So let's say Yeah, I guess it is. It would be easier to draw this as an animation to show you. But so here's our enzyme. A competitive inhibitor is something that's going to compete with the substrate for binding. Oh, and actually, I should probably label this right here, that part right here. It's called your active site. And so a competitive inhibitor and we'll make it like a little bit different. There we go. So it's going to have the same shape as your substrate, meaning that it's and by shape, I mean sequence of amino acids that allow it to stick to the active site. And so it's going to be composed in a similar way as the substrate, at least in part of it, because what it's going to do is it's going to bind to the active site. And when it's bound to the active site, your substrate can't enter. And so we call this a competitive inhibitor because it's competing with the binding of the substrate. Now, a non-competitive inhibitor, we're going to create a little variation to this enzyme, which enzymes do have, most of them they will have some kind of allosteric site. And so we could label this as an allosteric site. And we'll just say site that things can bind to that is not the active site. Just to define it real quick. And so let's say, you have another protein molecule, or actually, let's use a real life example. Real life. I keep saying real life. Let's use an example that we're going to see in nature, in our bodies, within our cells. Let's make the allosteric site shaped like a product. So it's like how do I do this? Boom. Okay, so this is, we can say that this is what is the type of triangle that is equal in length on all sides? Wow, it's been a while. So it's that type of triangle. And then so the allosteric site is going to be the shape of a product. So this product is going to have a tendency to bind here, hypothetically. And what happens is when the enzyme binds, not when the enzyme binds, when the, you could say, in this case, this product will behave as a non-competitive inhibitor. So we'll write that. So product can act as a non-competitive inhibitor because it may bind to the allosteric site of the enzyme, which will change the enzyme's conformation, preventing it from catalyzing the reaction. We could say catalyzing its reaction with substrate. It's a long definition, but so that's what's happening here. So this is what we would call feedback inhibition. So this enzyme, can freely bind to substrate, create product. But this is a way that our cells can check themselves. What if you don't want too much product? What if you want the perfect amount of product? Well, the best way to check this is if you start making an abundance of product, that product can bind to an allosteric site on the enzyme, preventing it from making more product. So it's just a check for itself. That's kind of like a 211 concept, but This will help you guys in the future. So that's feedback inhibition. You don't need to know that for the test, but know that the difference between a competitive inhibitor and a non-competitive inhibitor is that the competitive inhibitor binds to the active site, and the non-competitive inhibitor will bind to a different site. And that's going to change the conformation of this enzyme, preventing it from binding to the substrate. Sorry, that was kind of drawn out. Let's see, what's another hint that I can give you guys? Know the parts of the photosystem. You guys know the parts of the photosystem? Someone say that or write it out. There's 2 parts. Oh, okay. Someone wants me to just go over the citric acid cycle. Oh, someone answered the question. Photosystem. Antenna and what do we got? We got one more part. The antenna complex and one of you guys have to know this. Come on. Reactor complex. That would work. The reaction complex. There's another term though. Yeah, you could use reaction complex. You could also use chlorophyll A. It was the reaction center, but yeah, that works. You got the right idea. Just know that it's composed of 2 parts: the antenna complex, which funnels in that light energy, and then you have the reaction center, or chlorophyll A. You could just say chlorophyll A too. Okay, citric acid cycle. So this is something that I did post, and I hope you guys got the video where I walked through cell respiration. Have you guys gone through that or seen that? Okay, cool. I got 1 yes. I can go over citric acid cycle though. What are you struggling with in the citric acid cycle? Because if I go over the citric acid cycle, I don't want to confuse you with more information, because I can go into the citric acid cycle in detail, but a lot of the stuff you don't really need to know. You just the basic sub-steps. Okay. Okay. Basic stuff you need to know. All right. What you need to know is you got this 4-carbon molecule, and then we got this 2-carbon attached to a coenzyme A, and then you have this 6-carbon molecule here. Okay, so the basic concept is that pyruvate oxidation has occurred. You have this molecule called acetyl I want to make it black. Coenzyme A, acetyl-CoA. We have this molecule oxaloacetate. And acetyl-CoA is fused with oxaloacetate to form citrate or citric acid. And then you get the shuffling of atoms, you get the release of CO2, you have the production of some ATP, And importantly, you get the production of a lot of NADH and FADH2, which you bring to the electron transport chain. So I'll draw an arrow. What is going into this? What's going into this? If you're making NADH, right, you're going to probably start with NAD plus, FAD. If you're making ATP, you're going to start off with ADP, right? We don't even need to know numbers here. So that's probably what you're bringing to it. If I remember other stuff, I'll add it. And then what are you getting out of citric acid cycle? NADH, FADH2, ATP, CO2. You're creating CO2 during it. Yeah. Also, this is going to happen in the matrix. All this is going to occur, I'll just say citric. Occurring in the matrix of the mitochondria. And then also remember that, we know that these two guys are important because these are what we're going to use in the electron transport chain. And that's what's going to produce the majority of the ATP. Now, out of all of the cycles or all of the processes that happen during cell respiration, the one that's going to produce the most CO2 is your citric acid cycle. I might have a question like that. So I have a tricky question because my test is going to have some tricky questions. They're going to prompt critical thinking. So I want to prepare you guys for critical thinking. So let's say that you have some kind of animal like us, an alien, or we could just use a human. Sure, let's use a human. And they're born without mitochondria. So a rare disease with no mitochondria, okay? However, they find that the muscles still work properly. What do you think the result would be? Or why do you think that they can function, actually? The question to you is, why do you think that their muscles are still able to function despite not having mitochondria? So their heart's still able to beat. Yes, fermentation. So because they don't have mitochondria, which is responsible for the oxidative respiration, which is very efficient, well, they would instead undergo fermentation. So they'd probably start hyperexpressing all of the enzymes and things associated with fermentation. So they'd probably be really good at fermentation. And as a result, well, we know that fermentation causes lactic acid buildup. So it'd probably be pretty difficult and painful for the individual to move, but it would be able to move because it can. You can still produce ATP through fermentation. But yeah, so Liz is absolutely correct. You're going to get a lot of fermentation and therefore a lot of lactic acid buildup. Cool. Let's see, what else do I have that I can talk about? No, the what is going to contribute? It's I guess the reaction center in photosystem 2. Where is it getting its electron from? Water. Yeah. No volume. Oh, am I? Oh, you're speaking to Liz. I think she was talking to herself. Were you talking to yourself?
Yes, sorry. I was I
Okay.
was working through the steps in my head.
Yeah.
And yeah.
Yeah.
Sorry.
No, you're don't be sorry. You're absolutely right. Water is the correct answer. Where is photosystem 1 getting its electrons from? Photosystem 2. Perfect. All right. Where does the light reaction occur during photosynthesis? Yeah, you could say in the photosystem, but where? I guess. Yeah, you got me. Where within the chloroplast? Sorry, I should have been more specific. Liz got it right. The thylakoid membrane. Judah, you're still right. So it does happen. It does happen in the photosystem, but it happens within the photosystem within the thylakoid membrane. That was my fault. I should have been more specific. Where's the Calvin cycle going to occur within the chloroplast? Yes, in stroma. Okay, here's another question. So during fermentation, what molecule is oxidized and fed back into glycolysis? What becomes oxidized and fed back into glycolysis? This is going to be during fermentation in the mitochondria, or not in the mitochondria, sorry, in the cytoplasm, sorry, the cytoplasm of the cell. All right, we're getting a lot of waters. I'm gonna pull up the fermentation figure really quick. This is very important to know. Okay. Open image and tab. Can you see that figure? Okay, so we know that glucose enters the cell, and when it enters glycolysis, it produces 2 net ATP. And also as a result of glycolysis, you form 2 pyruvate. Also, another thing that you produce during glycolysis is NADH. If you're in the presence of oxygen, that NADH is shuttled over to your mitochondria and fed into the electron transport chain to produce lots of ATP, right? But if you're going to ferment, this NAD plus, which is converted into NADH, is then used during fermentation to produce lactate. What happens here is that NADH, or NAD plus, is one of the rate limiting steps in glycolysis. So glycolysis can't happen without NAD plus. And so what you're doing during fermentation is you're taking NADH and you're forming more NAD plus to feed that back in to glycolysis to produce 2 more net ATP. So fermentation on its own doesn't produce any ATP. The only reason why we say that it creates ATP is because when it's coupled with glycolysis will then be potentiated to go one more round and produce more ATP. So what is oxidized during fermentation? It is NADH. NADH is oxidized. So if NAD plus was oxidized, it would be something different. NAD plus, if reduced, becomes NADH. NADH, if oxidized, becomes NAD+. So that's definitely something that I'll ask about on the test. What becomes oxidized during fermentation and fed back into glycolysis? And know that it's oxidized too. It's important to know that it's oxidized. Okay. Know the formula for fermentation? No, that one's a little bit complex. Well, not necessarily complex, but I won't be testing on the formula for fermentation. I'll be testing on the formula for cell respiration, just the general formula for cell respiration, and I'll be testing on the general formula for photosynthesis. So what is reduced fermentation? So it depends on the species. If we're speaking about us, for example, so we make lactic acid, right? Pyruvate, believe it or not, pyruvate is actually reduced into lactic acid. Yeah, because if there's something oxidized, there's going to be something reduced. And in that case, it's pyruvate. Okay, here's one. What part of the mitochondria is highly folded? You have a part of the mitochondria that's very folded. Yeah, it's something that enshrouds the matrix, so it provides the barrier for the matrix. So you're on the right track. Liz says inner membrane. Yes, so your inner membrane is actually folded, and that's to increase surface area for the electron transport chain. So more surface area, more space where the electron transport chain could sit. So you're trying to increase surface area. Okay. What's the final electron acceptor in the electron transport chain of cell respiration? Yes, oxygen. Oxygen is the final electron acceptor in the electron transport chain for cell respiration. What is the final electron acceptor for part of the electron transport chain, or yeah, for the electron transport chain in photosynthesis? It's actually going to be NAD plus, or NADP plus, or NADP. You could just say NADP. Yeah. So when we're dealing with photosynthesis, they're going to use electron carriers, and I like to associate the P in NADPH with photosynthesis, or you could say plant. So it's like the electron carrier for photosynthesizers. So final electron acceptor in cellular respiration, or the electron transport chain in cell respiration, is going to be oxygen. The final electron acceptor in photosynthesis is NADP plus, or NADP. If you can't remember the plus, I'll get the point. Okay. And you guys remember, let's see if I can So I can find a quick figure. I guess it's not something I'm going to quiz you, or I can quiz you on, because I can't find a picture that's blank, but I'll just show you an image. So remember that the tonicity of solutions is going to refer to the solution itself. So if you have a cell, a group of cells, in a hypertonic solution, their water within the cells are going to leave the cells, and it's going to cause shriveling. If you have an isotonic solution, you're going to have an equal exchange of water. Same amount of water moving in is going to be the same amount moving out, so your cell's not going to be really changed in morphology. Now, if you have a hypotonic solution, this is why we don't inject people with distilled water, because you have less solute in the solution. That water is going to want to move into the cell because there's going to be more solute within the cell. And so it's through osmosis moves into the cell. And what happens if we're dealing with red blood cells is they'll burst. And so that's a case of, using distilled water. Now in plants, they have cell walls that provide them with structural integrity. And so even if their cell membranes expand, the cell wall will keep them from expanding too large to the point that they pop. So now it'll be different in things that have cell walls. And that's how you get, crispy lettuce, for example, or wilted lettuce, is if you have it in a hypertonic solution. Okay. Okay. So let's say you have an organism that has mitochondria. However, the mitochondria cannot use the breakdown products of sugar, like pyruvate, for aerobic cellular respiration. And instead, it could only break down things like amino acids. Why do you think that this organism, what do you think is wrong with the mitochondria? So you have a cell that can conduct fermentation, yes, and you could also conduct cell respiration, but you cannot conduct cell respiration for sugars, only fatty acids, only for amino acids. Damn, that's pretty good. Okay, you're so close. So Judah, that's an amazing answer. So it can't produce pyruvate. So it can still ferment, which means that you're going to need lactate still, but you're very close. What do you think could be wrong with the mitochondria? So you're still going to have pyruvate floating around because you're going to still ferment. And remember the mitochondria, it could break down and utilize other things besides sugars, besides pyruvate, right? Yeah, you're on the right track. Can break down water. Mitochondria can break down water. So what if your mitochondria, let's draw this out actually. So you got We'll make the mitochondria blue. Mitochondria is blue. And so if you remember, I talked about how, your mitochondria classically, we'll draw out, so glucose enters the cell. And then, glycolysis can happen. Glycolysis happens. You form pyruvate. Oops. Whatever. Okay. 3-carbon pyruvate, 3-carbon pyruvate. And in the absence of oxygen, you could ferment this, because in this case you're still seeing lactate. You're still seeing some lactate within the cells, and so we'll create lactate, and I'll label lactate because it's still going to be chemically similar but still different. So lactate. And now we also have amino acids. So we'll just say, make this green. We'll just say, these guys are amino acids. We know that amino acids are all going to kind of be different. And so some amino acids can be metabolized for energy and some cannot. But those amino acids can move, into the mitochondria, and we can get cellular respiration, aerobic, sorry. So we can still produce ATP from amino acids, but your pyruvate is never turned into acetyl-CoA. Your pyruvate, tries to get into the mitochondria and bounces off. What do you think the issue is? We still have lactate available, We have the presence of lactate because we have fermentation. We have pyruvate, which is just hanging out in the cell too, waiting to be fermented, because what else are we going to do with it? We have amino acids, and we have the production of ATP from amino acids. There's no pyruvate oxidation. So what do you have to do to get pyruvate to undergo pyruvate oxidation? You have to get it into the mitochondria. And so in this case, this mitochondria is lacking a transport for pyruvate to get in. Yes, pyruvate needs to enter the mitochondria. So no transporter. You're lacking the transporter. So that's another tricky question that you guys might see. All right. So in this case, it's critical thinking. You're like a doctor of cells in this case, Like you see the presence of these things, the mitochondria are still functioning, but you see a bunch of pyruvate and it's not able to metabolize pyruvate. That means that pyruvate can't get into the mitochondria to be converted into acetyl-CoA. Okay, that's probably the trickiest question on the test. And yeah, know that you know the equations for cell respiration, know the equation for photosynthesis, know what is oxidized in cell respiration, and know what is reduced in cell respiration. Okay, so I got a question. What would the thought process look like to figure this question out? Would there be photos or more information? So there wouldn't be photos, which I guess a photo could help. Actually, a photo really would help, huh? It would just be the words. It would just be, here's the diagnosis. You have an animal that is functioning normally. Well, I guess in the case that you can't transport pyruvate into your mitochondria, you're probably going to be pretty inefficient at producing ATP. So you might be a little bit more lethargic than normal. But lethargy, that would be probably something included. And you look at their cells and you notice that there's an abundance of lactate. And you notice that there's an abundance of pyruvate still hanging out in the cell. And you notice that their mitochondria are metabolizing amino acids for energy, or even fatty acids for energy. Yeah, so you go through the process, see what information is missing, and then you determine, well, why can't it metabolize sugars? And yeah, why can't it metabolize sugars? Well, because you're not able to. And I guess to be more specific, why can't it metabolize pyruvate? And the answer would be, in this case, because pyruvate is not able to be converted into acetyl-CoA, and it can't be converted into acetyl-CoA to be spliced into the citric acid cycle, because you're lacking the transporter to get it into the mitochondria. But this is going to be multiple choice. So, I mean, you use the potential diagnoses and you pick the best one. This isn't something that I would be like, okay, write me an essay on, how you would diagnose it. I wouldn't expect for you guys to, figure that out on your own. That would be probably more 200 level. So, but anyways, what is oxidized during cell respiration? Do we know? Glucose. I'm gonna write it out. Yeah, C6H12O6. So I'm just gonna write it out for you guys. So C6H12O6 plus O2. I'll put a space in between them. So you're making H2O plus CO2 plus ATP. So this is going to be your equation for cell respiration. And so the way I would draw this is C6H12O6 is oxidized. It eventually becomes CO2. It's oxidized. And then what is reduced? Yeah, oxidized gets smaller parts. So your oxygen becomes reduced. So we'll write so in cell respiration, sugar gets oxidized, your oxygen gets reduced. Now, amara, you put H2O, which you're close if we were talking about, or actually you're absolutely correct. Yeah, if we're thinking about photosynthesis. So let's draw that one out while we're here anyways. So in photosynthesis, what they're doing is they're taking light energy plus, or we just say light plus CO2 plus H2O to make C6H12O6 plus O2. And so in this case, what is being oxidized is your H2O. What is being reduced is your CO2. Does the order make a difference? No, it doesn't. As long as, you know, the stuff that's on the left is correct, doesn't matter the order, the stuff that's on the right is correct. Good question. That's reduced. Okay. That's the stuff that I have for you guys. If you guys have any more questions, I can answer those. Make sure you guys do the Kahoot. For sure, do the Kahoot. Highly suggest that. Not everyone took advantage of it last test, and I'm here to suggest, please take it. It's a good exercise. Okie dokie. Well, if you guys don't have any questions, that'll be the wrap up.